Here we go. This is the second chapter 9 section on the sarcomeres and muscular contraction at the molecule level, really. We introduce a part of the cell here, part of the muscle cell, the sarcoplasmic reticulum, a specialized version of the ER found in all cells that in a muscle cell functions to store and release calcium when it's called upon for muscular contraction. All the textual information on these slides is good here in this file. We discussed, again, to review, the thin filament is primarily actin, and the thick filament is specifically the protein myosin. The hockey stick, the thick filament, and the olives, the thin filament. Again, when the muscle is in a relaxed state, there's only a slight overlap. This occurs on the edges of the A-band. When calcium is released from the SR and the myosin is permitted to attach to the actin or bind to actin, and you get the ratcheting or sliding of the thin filament, that overlap increases, collapsing the H-zone, as we discussed. That's in the center of the A-band, in the center of the sarcomere. Read it, this is pretty clear. This ratcheting or sliding of the thin filament over the thick filament, again, it's the thin filament that slides or moves, not the thick filament. Eventually brings the Z discs closer together and shortens the overall length of the muscle. So, of course, you're wondering what stimulates the calcium release from the SR, and that's a nerve impulse. So skeletal muscle, being voluntary, must be told to contract, so it must be stimulated by a nerve. The nervous impulse or electrical current that is delivered is called an action potential. This leads to the calcium release, which leads to everything else that occurs during contraction. So we've got our sequence events here starting from 0 here. This would be the first event, an action potential. This would be the second event. Excuse me. This is the third event, I guess this whole thing here. Which follows what I want to call this 3A. Once the blocking agents are removed, myosin can now form a cross bridge with actin. What is that, 4? And 5, and last here is the cause of the sliding of the thin filament towards the center. Again, that's the M line there in the center. The calcium release is only temporary. It's quickly drawn back into the sarcoplasmic reticulum, which restores the blocking and allows the muscle to relax. We spent a little bit talking about how ATP works in this process. This is a visual where the text didn't come out, but maybe that's better. Here's your nerve. Here's your action potential traveling along the muscle cell membrane. The signal travels deep within the cell, stimulates the release of calcium from the SR, which binds to the blocking agents, causing them to slide over, allowing myosin to bind to actin, and then the myosin head pivots and slides that filament in, causing the overall length of the muscle to shorten. And this just zooms in and repeats that. The red little dots here are calcium. When calcium is present, it causes these blocking agents here to just slide over just a tad, allowing myosin access to the actin, which is here. So we talked about the cross bridge, which is the actual myosin to actin connection. When that head pivots, what occurs is called a power stroke. Then there's a detachment, and this is where we talked about rigor mortis. Upon death, the muscles are in a stiffened, contracted state for an hour or so due to the unavailability of ATP, which prevents detachment of the myosin from the actin. So you're stuck. Cross bridges are stuck together. That looks pretty. And here you see the pivoting and cross bridge formation. And here's our ATP starburst. And if you don't have ATP, you're going to stay here. And that's your rigor mortis again. This is interesting. We contrasted two kinds of exercise, anaerobic and aerobic, and the kinds of energy, a number of ATP that are generated by each kind of exercise. Anaerobic respiration is very quick, but generates only a few ATP available. This is a slow process and requires oxygen. Actually, is also important, but makes much more ATP. Again, you don't have to know much here. Anaerobic occurs fast and gives you enough energy for an explosive kind of event. This is your sprinter. And aerobic respiration requires oxygen, but can deliver you hours and hours of energy. And this is your marathon runner. The problem with anaerobic respiration, although it gives you energy much more quickly, it also builds up a unwanted waste product, which is lactic acid, which leads to muscle fatigue and muscle cramping. Aerobic exercise, which does not build up lactic acid, but requires, remember, oxygen delivery to your muscles. So a lot of the training in aerobic exercise is going to result in increasing the efficiency of oxygen delivery. So capillaries, more capillaries, which is going to bring more oxygen to the site, more mitochondria, which can burn that oxygen, and more myoglobin, which carries the oxygen. Whereas anaerobic exercise, and kind of maxing out, or a sprint, or a intense exercise that's only undergone for a minute or two. So what that's going to do, actually, it's going to tear the fibers. The intense stress the muscles under tears the fibers, and you get increased filaments or bigger muscles, hypertrophy. So my analogy was you don't actually increase the number of muscle cells when you exercise anaerobically, but the way you get bigger muscles is by tearing those muscles and stuffing more filaments into those cells. So my analogy was your Christmas stocking. You know, here's your guy who's not working out that much. Here's his filaments. And I'm not going to have more stockings, but I'm going to have a bulging stocking. Because I've been such a good boy this year. No, and I'm going to stuff that with more filaments. And that's going to lead to muscle hypertrophy or bigger muscles. Good luck.